Okay, good afternoon, everyone. So we will continue our seminar series here. And today we are pleased to introduce Souhayla Kodad, who will present characterization of Beldi Almond Ecotypes in Eastern Morocco. So Souhayla is a member of Professor Al-Amrani's group and is supervised by Professor Atiqa Himam, sorry if I mispronounce your name. Go ahead, Souhayla.
Thank you, Professor. I am honored to be part of this meeting today to present to you our research concerning the characterization of Beldi Almond Ecotypes in Eastern Morocco. This research is a part of a project which is titled PARA, who concerns almond in eastern region of Morocco, its biodiversity in post-harvest recovery, derivatives and byproducts, which is a contribution for the valorization of almond sector in eastern region of Morocco. So our main objectives are the prospection of local almond seedling population, known locally as Beldi, in eastern Morocco, and also pomological characterization to identify genotypic richness for biodiversity conservation and preservation of selection promising genotypes, as well as physical-chemical analysis of almond oils and co-products for improving farmers' incomes, and micropropagation of selected Beldi almonds ecotypes to be incorporated to new plantation projects, and finally, the verification of genotypic diversity of Beldi almonds and identification of relationship between Moroccan almond germplasm and some foreign cultivars. So for the first study, it concerns the evaluation of phenological diversity of Beldi almond. Three main zones will be concerned by our study, which are in Sfa, Sara, Rislan, where periodic visits were made till October, till the harvest, for the selection of trees based on several parameters, such as the age of trees. We mainly work on aged trees, abandoned, that don't receive any attention from local farmers and yet remain productive. So these visits allowed us to access to different phenological stages of Beldi almonds and allowed us to classify according to the BBCH scale, which is represented here by this figure, starting by the buds of winter, which is represented by the stage 00 till the harvest, or the development of fruit, which is represented by 72 stage. This study also allowed us to classify our genotypes to three main categories, which are the early blooming almonds, the intermediate ones, and late blooming genotypes. Most of the current studies are concerning the late blooming genotypes because they allow to escape to the frost of spring, which cause huge damage in almonds. But in my opinion, it's important to maintain all the categories to preserve the genetic diversity of almonds. We also opt for evaluation of different morphological characteristics of the almonds. We evaluated the morphology of trees and flowers as well as the leaves. So we used a chromometer, which is represented in the figure number A, to measure the intensity of colors. The colors of petals range from white to dark pink. We also measured using a clipper the several physical parameters such as the late and the lidded. So we also evaluated the quality of the fruits. For that, we used an international descriptor for the measurement of different parameters, such as the length, the width, and the thickness. Other parameters, such as sphericity, the geometric mean diameter, and the volume, were calculated based on mathematical formulas. For the almond kernels, the same parameters were measured, as well as two main added ones, which is the double kernel rate and the shelling rate. In fact, the double kernels are one of the main characteristics of Beldi almonds, which is the fact of development of two kernels and one nut. And it has, unfortunately, a low commercial value, hence the interest of its valorization by oil extraction, for example. We also evaluated an important parameter, which is the hardness of shells, because the almonds are, Beldi almonds mainly, are characterized by their shells that are very hard. So we did a compression test to assess the shell cracking load, and we classified our genotype from tender, hard to very hard genotypes. These shells represent more than 70% of the biomass of the nuts, which is a very important amount and can be valorized mainly for the protection of bioabsorbents. For that, I invite you to kindly check one of our publications by a member of our research team, which entitled On the Suitability of Almond Shells for the Manufacture of Natural Low-Cost Bioabsorbent, which represents an essay for production of low-cost bioabsorbents. A second study concerned the evaluation of the valorization of almond or double kernels by oil extraction. So we took the Beldi kernels and we extracted them by using the local cooperatives to evaluate both the quality of our oils and also the quality of the extraction process in the eastern region of Morocco. So we evaluated acid and peroxide indices, oxidative stability, fatty acids, and tocopherols. Also, the oil-pressed residue, which is the powder that we obtained after extraction, has been evaluated. We evaluated the dry matter, the total protein, the crude cellulose, and the mineral content. We obtained an important results showing a high quality of the produced oils and cakes. And I invite you again to check one of our publications entitled Evaluation of Pomological and Biochemical Quality of Moroccan Almond Native Genetic Resources for Conservation of Biodiversity. So our third study concerns the micropropagation of promising genotypes by in vitro tissue culture. So we opt for a culture of the age trees by using in vitro tissue culture. We use the nodal herbaceous cutting, and we cultivate them on different types of media and using different plant growth regulators. We also use leaves for regeneration of explants, and the obtained shoots from both processes were rooted and successfully acclimatized to in vivo conditions. And I invite you again to check our recent publication concerning the effect of culture media and plant growth regulators on shoot proliferation and rooting of internode sections. And currently, which is the recent study that we are trying to create, which is the assessment of genetic diversity in phylogenetic structure through SSR markers. So we selected the genotypes in 3 main studied zones based on the previously described parameters, the phonological ones, as well as the physical chemical ones. And we opt for a DNA extraction using a CTAB buffer. We selected 7 main microsatellites for comparing the studied genotypes. And we're looking forward to compare our results by mainly comparing the observed heterozygosity, discrimination power, as well as the polymorphism, to evaluate as well as to prove this morphological diversity by genetic one. So as a conclusion, we can say that this study can be completed by choosing a wider range of genotypes in tissue culture techniques with primary focus on somatic embryogenesis. Also, we can evaluate other functional properties and biological activities of local almond, and selecting self-comparable and late flowering ecotypes, and finally incorporating these genotypes in new projects and new plantation projects. And thank you for your attention.
Okay, thank you very much for this presentation. I would say that you went very fast. So it's a fast-paced. So next time when you present, please be a little bit.
I did my best to make it.
Take your time. Yeah, we have 1 hour. I mean, we say presentations could be 20 to 25 minutes, but that's fine. That's fine. Yeah. I would ask if there are any questions from the audience. Then I have some questions, but I will be last. Anybody? Any questions? I see silence here. Okay. Okay, I will ask some questions to. Yeah, I didn't see. Did you first? I have one question about the quality of seeds of these this Beldi. Did you do any studies on the quality of?
Yes, I did the study on the almond nuts was basically on the rentability of almonds, but I didn't do it exactly on the almonds, but I did it on the oil extracted from these almonds. So I evaluated the quality of the oils as well as the case obtained. So in the results obtained showed the very high quality of the obtained both the obtained oils and cakes, showing high values of tocopherols and as well as fatty acids. And for the cakes, they are very rich in fibers and as well as minerals. So they can be used as in cosmetology or even in food industry.
Very good. Since you talked about tocopherols, do you know what's the quantity of tocopherol in oil of this Beldi compared to other genotypes that you studied?
Yes, compared to these, they showed significantly higher results, ranging between 560 milligrams per kilogram. Excuse me.
500 ppm, exactly.
ppm, okay.
500 ppm.
Okay, 1500 ppm of tocopherol. Okay, good.
I have a question, Majid, if I can interrupt yours, and then since we're talking about tocopherols.
Go ahead.
So how did you do your phenotyping for tocopherols, and what type of tocopherols did you measure? Did you measure the alpha, beta, delta, gamma?
Yes.
So which ones?
The major element in the almond oil is alpha tocopherol, followed
So you said the alpha tocopherol.
Yes,
is the major. The
major.
Major.
So how much of the 500? You said you had a total of 500 milligram per kilo, yeah? So how much of that is alpha tocopherol?
ppm.
ppm, they did it.
ppm or milligram by kilogram.
Yeah. So how many of that is alpha tocopherols?
It's around 400, I think. It's around 400 or more than 400. Is the major.
Is the major element.
Yes. So how many grams of almond you have to eat to get that, let's say to get 100?
You have to ask this question to Californian producer.
But in dietary, the recommendation is like you should not exceed 40 almonds per day. So it will be largely enough if you have already a good alimentary.
But there is a deficiency in vitamin E in Morocco for a lot of people. So this would be a good recommendation. You can so it's easy to just buy it from there and then get them their vitamin E intake.
True.
Yes, but it's too expensive for everybody to buy almonds.
Well,
did you know that we have almonds from California in Wijda, and it's cheaper than our production?
Yes. Yeah. So you have an HPLC machine in the lab that you use for tocopherol?
Yes.
Yes, it's under the responsibility of Professor Sarghani.
Okay. So maybe I have also a project in the lab about tocopherols, but in soybean. So maybe we should talk. If you guys are interested, we can probably do some work together. In that.
Yes, we'll be glad.
So how much it costs you to do the HPLC per sample, for the tocopherol?
No, 1 gram will be largely enough.
No, the cost for running
the cost of analysis.
I thought the amount of
we can't give you an idea, really.
Okay.
We don't have an idea how the cost of one analysis. We don't have an idea. We have the machine in the lab, and we use it for triacylglycerol and for tocopherol, but we don't have an idea on the price.
Okay. Yeah. Well, if you guys are interested, so we can probably talk later. We can have a Zoom, and then I can explain to you what we do with the tocopherols in soybeans. Yes. And then we can maybe do some kind of collaboration project on that. So that would be a win-win situation for both of us.
Thank you very much for the idea.
Yeah, I see Belhaj also have a question.
Yes,
I
yes.
think. Khalid, do you have still questions, or you can move on?
Let Belhaj go, and then I can ask later. Okay.
I thought he. Yeah.
Thank
go
you
ahead.
very much, Professor Abdelmajid. Firstly, I want to thank you for this scientific meeting. And secondly, I have one question for my colleague Souhayla about the specificity of this ecotype in comparison with other varieties introduced in the eastern region of Morocco. Thank you.
Thank you. Thank you for your intervention, Mr. Kamal. So as I previously said, it has a higher tocopherol content, and it presents also a richness in oils. It's very rich compared to the other studied genotypes, which are introduced here, which is favorable for many uses, mainly for bakery, because they prefer mainly the almonds that are very rich in oils. And also it has an important character, which is lower. It presents a lower price compared to the introduced cultivars, so it will be easier to get it.
In particular to Ferranis and Ferradiel varieties.
Firstly, as I previously said, so it has the higher tocopherol contents, and it has a higher oil content, and also it's cheaper. And what else? It doesn't need any attention from local farmers. While for Ferranis and Ferraduel, they have to be treated for years to be productive.
Hey, thank you. Thank you very much. I have invited you to discuss with Reda Milhawi about the characteristics of Beldi ecotype, in particular the difference between other varieties in fatty acids profile and glycerol profile.
Thank you.
So I have a question. I have a question actually. So we hear a lot, Moroccan consumers, they always say, lose el Beldi, lose el Romi. So is there really any differences between the almonds that you find, the ones that are native to Morocco, or, not native actually, the one that has been grown in Morocco for many, many years, because almond is not native to Morocco. And the other, California almond or European almond, is there any differences, in terms of, I would say, nutritive values and also nutraceutical values?
Yeah. So for the nutritional values, both of them are very rich in all the previously described parameters. But for us, why do we prefer it? Mainly for its low price. And also it has a small shape, so compared to the introduced cultivars, they are mainly present in bigger diameters, so they are more presentable to be used in important events or even to be presentable to guests and stuff. But while for the Beldi almond, it generally has a small sizes, it has darker colors, so it's less presentable. So it's mainly used for a bakery, or because as I previously said, it's very rich in oils, etc. So this preference is mainly related to the price as well to the interest of having a lot of oil. So the past, it maintains more in bakery and stuff. Well, and also it has a good taste, represented by a little bit higher sugar content, we can say. Mm-hmm. It's preferable for us. And also for the people who prefer the taste who's between the sweet and sour, not sour, but it's a little
bitter.
Bitter.
Mm-hmm.
So this, due to the pollination of sweet almond, but a bitter one. So we have the taste that differ a little bit from the normal almond, sweet almonds. So there is some people that prefer that.
Okay. So in the US, we have a lot of almond milk actually, and it's mainly designed to people that do have that are lactose intolerant. And I know that the population in Morocco is almost 95% lactose intolerant. Mm-hmm. So do you see any applications, in the future for producing milk from almonds in Morocco?
Yes, it's a very low cost, feasible technique that can be used everywhere. So it's just it just needs to be aware of. So I think it's very possible to be that Beldi almonds mainly can be used for production of almond milk.
Okay.
But its taste, Khalid, is not like regular milk. So
Yeah.
I don't think, I tried it myself. I didn't like it that much, but
for the Moroccan taste, I don't think that it will work. They don't care about the tolerance for lactose.
They just drink.
Okay.
They care more about the taste of milk, I think.
Yeah.
But
Yeah, I have.
be possible
Go
for
ahead.
sure.
I personally, I like, almond milk drinks, So the one that they have in Morocco is, but they mix it with milk. So in any case.
Yeah, I have one last question about the tissue culture. So I saw that you tried to propagate this ecotype or Beldi in tissue culture. My question is, did you reach a conclusion that this plant can be actually, you can reach the goal of being stable and fertile in the field, or you are not there yet in your studies?
Not yet.
Not yet. So what's your stage? You have like plants, whole plants after in tissue, but you didn't transfer them yet to the greenhouse or the
They are in the greenhouse currently, but they're not productive yet. So
they are not. They don't reach.
They need at least 2 years. Yeah, they need at least. I am in my second year. No, a third year today. So they need at least 3 to 4 years to be productive. So we can compare both. But we are trying to compare the genetic stability of the obtained in vitro culture plants. So we'll be comparing them to the mother plants to see if there is a genetic stability or not. I didn't do it yet, but I am working on it.
So you're not going to get your PhD until we see the first almond coming from the tree, yeah?
From tissue culture.
It's already there. Just need to taste it.
Okay, great. Any other questions from the audience?
Well, thank you very much. Thank you.
Good luck.
Thank you so much.
Good morning or good evening, doctors and everybody there. I am Atiqa Mihamo, the supervisor of Souhayla Kodad with the whole team. I want to thank you for the opportunity for Souhayla to do this presentation. What I would like just to see, because with with Beldi almonds in the region, there is a big program to distract or to replace the whole old by new trees because of this disease. So what we are doing could be just how to say that we are in a big deal with the Ministry of Agriculture here because we have some ecotypes, very interesting ecotypes, but they are going to track them, so we will be in trouble to compare we get from these trees after tissue culture to compare them to their mother plant. So we hope that we will have enough time to get this comparison and to save some ecotypes from this diversity of almonds in this region and in other regions in Morocco. It's a big program and a big problem of with the almond population or ecotypes in Morocco. So I want to give this precision about this problem. Thank you.
Thank you very much.
Thanks.
Yeah, this question is more for the group leader, Dr. Al-Amrani. Did you think about doing some QTL mapping in almond, or do you have populations that you can do QTL mapping on that, or you never thought about this?
To answer this question, that's what my colleague Atiqa said, that in Eastern Morocco, we have a big program. It's named in French ProFAO. This program have been supported by Belgium Kingdom, and they introduce new species in this region, specifically Ferranius and Ferraduel. And they go with the idea to have profitable plantation. And they put some cooperation with other laboratory, and everybody go to new plants of Ferranius and Ferraduel. And one time we discussed in Professor Atiqa, which look for the biodiversity. She chose to have this idea and to work on Beldi almond for two ideas. First one, the biodiversity. As you know, it's not easy for a small group like our group to resolve this problem of biodiversity of almond in our region. But the second aspect, it's an economic aspect, and I lead this aspect with Beldi Almond. We try to show to farmers that if Beldi Almond gives kernels which are sometimes not really profitable, because kernel are double, kernel are have small shape or small form. Sometimes they are, I don't know how to tell it in English, kernel are not homogeneous. So we have two aspects. We have this aspect, how can we use Beldi almond kernels and go to have an idea with a profitable for farmers, and try to tell them that you have to valorize or to evaluate this kind of culture, because it's really resistible to drought and some everything. And in parallel, we have Ferranius and Ferraduel. So we have to compare the two type of culture. And if we are going to in the aspect of genetic to QTL, at the moment they try the SSR, but for QTL I don't know. For the moment in my group, they are going to make some work like this.
Okay, yeah, because I just read some articles on that, because through this you can introduce some important agronomic traits that you just talked about, like kernel and the size of the seed, etc., and improve them through marker-assisted selection, etc.
I invite my colleague Atiqa to take note about this idea, and if we have to be in contact with you on this task, we will do.
Okay, good. Thanks. I see Usama have some questions. Go ahead, Usama.
All right. Thank you for your presentation. I'm sorry I'm here late. I just got off work. You were talking about those Beldi almonds. They are sweeter than the rest of the varieties. Did you do any cyanide? I don't know, profiling on it, because the cyanide is what gives it the bitterness, and you can compare that to other profiles that will allow you to introduce that Beldi variety in breeding programs, and that will attract more people, more attention to why those Beldi varieties are good to be introduced either in breeding programs, or we can use them to introgress other characters that we need. And Abdelmjid was talking about Q-tails. There is a really fine reference genome of almonds published there, which Q-tail mapping should be a really good idea to do. And one more question. There is any national willing to have a reference genome for that Beldi variety so we can protect it from, there's a lot of Spanish industry trying to, they've been in that region of Morocco for a little while and trying to get those nice genotypes. So a national program to protect that landrace or cultivar, whatever we can name it, should be very interesting. Thank you.
Thank you for your intervention. In fact, no, I didn't do this kind of test, but I'm looking forward to do some of if it's worth the study. While for the preserving the genotypes, etc., I know that they do exist some works from the International Institute of Research in Agronomy in Meknes, I guess. They preserve some interesting genotypes, and they do use them, and they compare them to other foreign and introduced cultivars. But I don't know if it's one of the main interests of our country, or just the research made by the institution.
Okay. So if we do not have any questions, we thank you.
I have one, Majid. One question.
Go ahead.
So are you, or did you think about maybe some breeding programs in Morocco where you can cross the Beldi almonds with, let's say some of the best California, cultivars?
Not on the state at the moment.
Okay. So Osama mentioned, that there is actually a draft genome for almonds. Now it's actually going to be easy to do a mapping, if you are developing populations or anything, so you can just take advantage of that draft genome to develop good, markers that you can, trace your crosses and trace some of the traits that are of an interest to you.
Yeah, actually, in our laboratory.
I think first of all, we have to do some genetic assessment. And then we can try to find a CTAB program for the Beldi ecotypes.
Okay.
It is in progress. It's a perspective for us, but for the moment, we are not a good equipment in molecular biologists. We have some cooperation with medicine faculties and some cooperation with stranger laboratories in Germany. And we are trying to, first of all, to do some genetic assessment, and then to find some good genes to do cross breeding with others.
Yes. Well, one type of collaboration that could be done, between us, the Moroccans that are here, and you guys, if you can actually have a nice collection, of ecotypes of almond in Morocco, we can try to do some sequencing on those ecotypes. And this way, we can teach you how to do some clustering. This way, you can at least have a nice representation of what is in your cultivars. And then when you think about breeding in the future, or maybe protecting some of the cultivars, at least you have some molecular tools and some sequences to base your analysis on.
It's an excellent idea, and it is very good for us. So she has to attempt for a Fulbright scholarship to do this in your laboratories. It's a good idea for us.
Okay. I mean, with or without the Fulbright, if you can start, collecting at least, the different ecotypes and having, some
DNA. Already done. They are already done.
Okay. How many different ecotypes you have now in Morocco?
More than 100.
100.
More than 100.
Okay. And those have been characterized. You have the phenotypic characterization and the location of every one of those. Yeah.
Yeah. Yeah. All of this is done.
Okay. Well, maybe we should have another Zoom where we can discuss probably, you know, how we can move those ecotype into sequencing and then the cost involved in it, and then how we can raise that money to do it, because that's going to be, you know, the best thing to do for the ecotypes of Beldi that do exist in Morocco.
Yeah, we agree the ideas, and it is very interesting for us.
Okay.
Very good.
Very good. Osama, are you still there?
Very good.
I am still here. I was listening. Now this is interesting to me.
Yes.
Yeah,
well,
like what I think we should apply. Morocco is like a reservoir of all land races and germplasms that have been adapted to biotic and abiotic stresses for hundreds of years. We should apply some genomic and protect those, because the French, they came in, they stole everything. The German came in, stole everything. But now we have some stuff that they need to be protected. And all we have to do, since you did the phenotyping already, we should just start drafting a proposal so we can get that money to genotype those by sequencing and then protect them. Or I don't know, whatever you guys want to do with that.
Would you please let us your references? Then we could talk with you later. We are interested by the idea.
Okay.
Very good.
Yeah, I think what we will do is we can organize maybe another Zoom meeting during the week, either next week or the week after, where we can, you know, discuss the idea further. And we can probably put the first plan where we can, you know, we here in the US, we can put some money to do some sequencing, and then train some of the people to start looking, you know, at the correlation of those sequences with some of the phenotypes that you have. One thing that we can do, for example, for those 100 accessions that you have or cultivars, if you can phenotype every one of them, for example, just for like tocopherols, and then for another trade, you can do like just 3 trade, you know, in those cultivars. So we can actually look at the association. We can do some genome-wide association between what you have and the trade that you have that will justify, you know, in the future going with a nice proposal to one of the agencies and asking for money to do, whether if you want to develop, you know, breeding programs for it, or you want to develop some varieties. But if you just go like this and you tell them I have this, nobody's gonna, you know, look seriously and favorably. What the others, they will try to do, they will try to take advantage of you and take those accessions from you and then use them in their own stuff, you know?
Yeah.
Okay.
Yeah. With pleasure, with a big pleasure.
Thank
for
you.
this meeting.
Good.
Okay.
Well, maybe Majid, we can do a follow-up, you know, with another Zoom. We can, you know, look at the availability of the whole team the week after, maybe next week. And then we can have a 1 hour Zoom when we can talk about these things. Yeah?
Okay, yeah, absolutely. That's actually our goals at the Council, is to increase collaboration between the US and Morocco.
Genotype.
Okay. Thank you.
Okay. Thank you again. We thank Souhayla and Dr. Al-Amrani and Dr. Mihammou's group for this wonderful presentation. And we will continue next month with our last presentation, actually, from your group as well in this academic year.